Welcome back, everyone. I was thinking about uh, finishing the immune system with the circulatory system, basically the blood and hematology, and also if there is anything with regards to lymphatic system, we can study all of them together, and then we can do practice questions later on all of these subject matters. So let's start with the immune system. I believe we have 86 flashcards for this, so let's go over them. So this is from last night where we left off, and it's basically about innate immunity. We have two types of immunity: the adaptive immunity and the innate immunity. And the innate is non-specific, and it's always active. And the examples are skin, mucus, stomach acid, and tears. So you can think of your primary barriers, your first line of defense. Skin is common sense. Mucus, think of when you have a flu and your nose is running and it's watery. You have the mucus that is helping you to fight the battle, and you have stomach acid, which is always there to make sure that it can kill any harmful materials, substances. And you also have your tears in order to um, in case you have any allergic reactions, for example, right? Now, as you can see, the right side here, it is innate immunity. The only two cells that are shared between innate immunity and adaptive immunity is the Y8 T cell and the natural killer T cell. But we should keep in mind that the natural killer cell is part of the innate immunity, as well as the mast cell, the dendritic cell, the macrophage, the complement protein, neutrophil, eosinophil, and basophil, which are granulocytes. So we can see that we have a cellular adaptive immunity and the humoral one. And the humoral immunity is all with regards to B cell immunity, but the cellular immunity is with regards to T cell immunity. We have the hematopoietic cells, which are in the bone marrow, and these stem cells give life to the myeloid progenitor cells or the lymphoid progenitor cells. So the lymphoid progenitor cells give way to the lymphocytes, and the myeloid progenitor cells give way to red blood cells, platelets, the granulocytes, the monocyte. So it's all with regards to innate system. But for now, we will go to the next topic. We have here the adaptive immunity, which are defenses that are specific to the invader. So we have cell-mediated immunity and humoral. Humoral is B cells and cell-mediated is T cells. We have CD4 plus T cell, which have MHC2, and the CD8 plus T cell, which have MHC1. And the rule is to know that the multiplication of CD4 times 2 is 8, and CD8 times 1 is 8. So it should all accumulate to 8, so that CD8 plus has MHC1 and CD4 plus has MHC2. What is a physical barrier and secretes antimicrobial enzymes like defensins? Skin, I believe, is a physical barrier which secretes antimicrobial enzymes like defensins. The skin serves as a physical barrier to prevent passage of many disease-causing microorganisms. The skin is also slightly acidic and has good bacteria. So here we have intact epithelial barrier. Remember that in capillaries we had endothelial cells, but here it is epithelial cells. And so now we can assume that anywhere that you have thelial is referring to a barrier, right? And now our epithelial barrier is damaged. So you can see there is an initiation of Th2 immune response. There's the sensitized host, and there's clinical outcomes like atopy and asthma. That is made in your nose, mucus that is made in your nose, throat, and lungs, traps bacteria, viruses, and dust. It traps bacteria, viruses, and dust. In respiratory system, mucus is propelled upward by cilia via mucociliary escalator. So we have a mucociliary escalator that propels mucus upward by cilia. So it is cilia that have the mucociliary escalator. The first step is that the cells produce mucus. So the question is, what type of cells produce mucus? Let's go to the immune system we have here, right? So let's start here. Immune system. What type of cells produce mucus? Then the bacteriophage form bonds with sugars in mucus, and then a protective layer is formed, and then bacteriophage reproduce and attack on their own. So that is what is the scenario here. What is an antimicrobial enzyme that forms part of the innate immune system? The innate immune system is about non-specificity. So what is an antimicrobial enzyme? I believe it is, as we saw, it was defensins, but here the answer is lysozyme. It is found in tears and saliva. Lysozyme weakens the bacterial cell wall, allowing it to rupture, killing the bacterium. So, as you can see here, lysozymes, they're found in tears and saliva, and their job is to break down the cell wall of the bacteria. So it is an antimicrobial enzyme, and it's part of the innate immune system. What system is a part of the immune system that enhances, complements the ability of antibodies and phagocytic cells to clear microbes and damaged cells from an organism, promotes inflammation, and attacks the pathogen's cell membrane? I believe it should be the lymphatic system. No, it's the complement system. So the complement system is part of the immune system. It enhances complements the ability of antibodies in phagocytic cells. So it helps the antibodies and phagocytic cells to clear microbes and damaged cells from organism. So it's like a recycling job. It promotes inflammation. It attacks the pathogen's cell membrane. It attacks like the cell membrane of the bacteria or the virus, right? The complement system consists of 3 pathways. There is the classical pathway, the alternative pathway, and lectin pathway. So this is the classical pathway. The microbe with antigens comes into our cells, and then you can see that we have antibodies for the antigens, and there is the degranulation of mast cells, and we have opsonization and cytolysis. We need to know more about these. So the complement system consists of neutrophils, monocytes, mast cells, macrophages, dendritic cells, B cells, T cells. It helps the innate immunity. For example, opsonization, lysis of pathogens, chemotaxis, inflammation, and cell activation. It also helps with disposal system, clearance of immune complexes and apoptotic cells, and also augmentation of antibody response, promotion of T cell response, elimination of self-reactive B cells, and enhancement of immunologic memory. Okay, we have these terminologies for the complement system and the innate immune system. So we will come back to it if it's not mentioned here in the flashcards. But for now, we just need to know the complement system consists of the three pathways, which is lectin, classical, and alternative pathways. And we just need to know that the complement system complements the innate immune system, and it helps with inflammation. It helps with complementing antibodies and phagocytic cells to clear microbes and damaged cells from an organism, and it attacks the pathogen's cell membrane. So, a type of protein interferes with viral replication and dispersion. What is a type of protein that interferes with viral replication and dispersion? I believe it's the antibodies, or it's interferons. So interferons interfere with viral replication and dispersion. It is given off by virally infected cells. So surprisingly, what is referred here is that interferons are given off by virally infected cells. Oh, so the cells of our body that are already infected with uh, viruses, viri, however you call it, they're the ones who give off interferons to interfere with the replication of the viruses. So as you can see, we have a virus infection, then there is the viral assembly, and then there is the activation of the INF gene, and then the INF is released out of the first cell. So INF is interferon, right? It's the interferon gene. And then at the same time, the viral assembly gives out new viral particles, and then the virus infecting another cell. But here, the INF binds to the receptors, so it gives a signal to nucleus. It might actually be kind of a GPCR, the G protein coupled receptor chain of reaction. So it can give a cascade signal to nucleus, and the nucleus produces antiviral protein, which destroys the viral particles. There's a YouTube video on this, so we can watch this later perhaps. What does ingest pathogens? Break them down into antigens, then present those pathogen specific antigens on MHC2. It's macrophages. So macrophages ingest pathogens, they break them down into antigens, and then present those pathogen specific antigens on MHC2. So as you can see here, there is a macrophage that is digesting, phagocytosing, whatever you say for whatever the verb is for phagocytosis. It's phagocyting the foreign microbe with antigens, and then the macrophage ingests antigens. It processes them and presents them to the T cell. That's where you transfer to the MHC2. And there's a note here that macrophages do ingest pathogens, but it then breaks down that pathogen into smaller pieces of identifying proteins called antigens. Those antigens are then presented on the surface in the context of MHC2. The difference between pathogens and antigens is really important, especially when it concerns MHCs and antigen presenting cells in general. So again, macrophages, they do ingest pathogens, but it then breaks down that pathogen into smaller pieces of identifying proteins called antigens. Those antigens are then presented on the surface in the context of MHC2. The difference between pathogens and antigens is really important since it concerns MHCs. So it is antigens that are on the MHC, but pathogens are microbes or viruses that the macrophages ingest, and they ingest their antigens. We have the mast cell. It dilates blood vessels and induces inflammation through release of histamines and heparin. It recruits macrophages and neutrophils. It is involved in wound healing and defense against pathogens, but can also be responsible for allergic reactions. Then you have macrophages. They are phagocytic cells. They consume foreign pathogens and cancer cells, and they stimulate response of other immune cells. Then we have natural killer cell. So these are all adaptive immunity, right? Innate immunity. So the location of the mast cells for inflammation basically is in connective tissues and mucous membranes. For macrophages, it migrates from blood vessels into tissues. Then we have natural killer cells. They kill tumor cells and virus-infected cells. And then their location is in blood, and they can migrate into tissues just like macrophages. We have the dendritic cells. They present antigens on its surface, thereby triggering adaptive immunity. So dendritic cell is an adaptive immunity cell. It presents in epithelial tissues like our skin, including skin, lung, and tissues of the digestive tract. So our skin and lung and digestive tract have epithelial tissue where dendritic cells are presented there, and they migrate to lymph nodes upon activation. They can also go to our lymph nodes upon activation. Then we have monocytes. Monocytes differentiate into macrophages and dendritic cells in response to inflammation. So when we have inflammation, monocytes can turn into either differentiate into either macrophages or dendritic cells. They are stored in spleen, and they move through blood vessels to infected tissues. Then we have neutrophils. So neutrophils they are first responders at the site of infection or trauma. This abundant phagocytic cell represents 50 to 60% of all leukocytes. They release toxins that kill or inhibit bacteria and fungi and recruits other immune cells to the site of infection. They migrate from blood vessels into tissues. So neutrophils is like the majority of our leukocytes. So 50 to 60%. Then we have basophils. They are responsible for defense against parasites. They release histamines that cause inflammation and may be responsible for allergic reactions. And they circulate in blood and migrate to tissues. And we also have eosinophils, so all the granulocytes migrate from blood into tissues when needed. So they circulate in blood and normally, and they go to tissues if needed. And eosinophils release toxins that kill bacteria and parasites, but also cause tissue damage. So that is so far. So if needed, we will make flashcards on these. I think we need to. So we will do that. Keep it in our list. I'm just writing it in my notebook to do it. We had also the complement system. So for now, we just need to know that macrophages ingest pathogens and break them down into antigens that they represent as on MHC2. So we have here glycoproteins are present on all nucleated cells. Glycoproteins are present on specialized antigen presenting cells, APCs, including macrophages, dendritic cells, and B cells. So I believe MHC2 is the second one, and MHC1 are present on all nucleated cells. That is right. So MHC is major histocompatibility complex. So major M histocompatibility complex. And here we can have a visual representation of MHC1 and MHC2. So they both have antigen binding clefts. They're like receptors, right? So we can ask, are MHCs receptors? And we know that they are glycoprotein, right? So because they are glyco, they can be inserted. They are inserted into the phospholipid bilayer, and because they are protein, they can be a receptor here, right? They have a beta 2 micro. So MHC1 has a beta 2 microglobulin. So it has alpha 1, alpha 2, alpha 3, and then one beta 2 microglobulin. But MHC2 has alpha 1 and alpha 2, and beta 1 and beta 2, just like hemoglobin, if you remember. Then we have this on this side of the cell membrane is the cytoplasm. So that side is the extracellular fluid, ECF. And here we have all nucleated body cells. And MHC2 is for macrophages, dendritic cells, and B cells. So MHC1 is for between the cancer cell and a CD8 plus T cell, but MHC2 is between the macrophage and a CD4 plus T cell. We can watch the YouTube video on this later too. So flashcards to that, YouTube. videos to watch. MHC2 and 1. So since we got this, we can put a good for this. What displays exogenous antigen to helper CD4 plus T cells? So it should be MHC2. So MHC2 displays exogenous antigen to helper CD4 plus T cells. Major histocompatibility complex 2. Because it's 2, we can also remember that it has 2 different subunits. And they're equal, so alpha 1 and 2 and beta 1 and 2. But for MHC1 it was alpha 1, 2, 3 and 1 beta. So that's probably the T cell receptor. And then you have endocytosis, enzymatic digestion, autophagy, fusion with Golgi, and then MHC2 class, and then lysosome. We also got this. So the main function of what cells is to process antigen material and present it on the cell surface to the T cells of the immune system. They act as messengers between the innate and the adaptive immune systems. On the cell surface to the T cells. So I believe it is the dendritic cells. Okay, we did not know this. So dendritic cells are the messengers between the innate and adaptive immune system. The main function of them is to process antigen material and present it on the cell surface to the T cells. So we have epidermis and intestine and blood vessels. And in the blood vessel, we have immature dendritic cell. So when there's a pathogen, antigen is uptaken by endocytosis, phagocytosis, and macropinocytosis. And then the mature dendritic cell has an antigen presentation to the T cell and cytokines and chemokines and co-stimulatory molecules in antigen presentation and T cell activation. So the question is, what are chemo and cytokines? What does kine refer to as the roots? Function. How do they interact? How do they interact with dendritic cells? Okay. So the main function of dendritic cells is to present the antigen on the to the T cell. So they process antigen material and present it on their cell surface to the T cell. Innate immunity are defenses that are always active and they are non-specific. It's like skin, mucus, tears. What is a type of lymphocyte that can recognize and kill cancer cells or other cells that have been infected? So it's the natural killer T cell. Natural killer cells are a type of lymphocyte. They recognize and kill cancer cells. Natural killer cells do not attack invading organisms directly, but instead they destroy the body's own cells that have either become cancerous or been infected with a virus, apoptosis. So they kill the cancerous cells of our body, the cancerous ones, or the ones that have been infected with a virus. So apoptosis is also what this refers to. These cells kill when there is no MC1 and when the cell is giving off stress proteins. Both have to occur, I guess. What is MC1? God knows. What is MC1 in the context of natural killer cells killing cancerous cells when there is no MC1 and when a cell is giving off stress proteins. Inhibitory ligands, no NK activation. Activating ligands, NK activation in cancer cell death. Natural killer cell, no natural killer activation. Natural killer activation in cancer cell death. We have a natural killer cell. Then we have the target cell. All these different scenarios. Then we have characteristics of target cell. Normal MHC1. No activating ligands. Outcome, no response. No MHC1. No activating ligands. No response. No MHC1 activating ligands. Attack target. Normal MHC1 activating ligands. Dynamic equilibrium. What is dynamic equilibrium? Does it attack it or not? So this is attacking it. Inhibitory ligands, activating ligands. I'm writing down to revisit this Khan Academy link for natural killer cells. Okay. What is an antimicrobial enzyme that forms part of the innate immune system? That should be the um, an antimicrobial enzyme. That should be lysozyme, right? So the difference between lysozyme and defensins is that they are both antimicrobial, but that's a good question. What is the difference between lysozyme and defensins? If we browse here, where are we? Let's see. We will figure that out later. What was interferon? Interferons interfere with the viral replication and dispersion. Okay, so for this, we can put it as hard. What system is a part of the immune system that enhances, complements the ability of antibodies and phagocytic cells to clear microbes and damaged cells from an organism? They promote inflammation and they attack the pathogen's cell membrane. So it is definitely the complement system that does that. It has the classical pathway, the lectin pathway, and the alternative pathway. You need to understand these. So, what are the similarities and differences between classical, alternative, and lectin pathways of the complement system? Humoral comes from Greek word for fluid. Immunity is immunity provided by. So humoral immunity is provided by antibodies. Kills antigens while they are floating around in the fluid. So while they are fluid in the these antigens are in the fluid in our blood, the antibodies kill them while they are floating around in the fluid. So on the left side, you can see that we have the humoral antibody-mediated immune system that control of freely circulating pathogens. the extracellular antigens. So a B cell binds to the antigen for which it is specific, usually requires cooperation from helper T cell. Then the second step is that the B cell, often with stimulation from helper T cell, differentiates into a plasma cell. So first it gets the help from T cell, binds to the antigen, and then the B cell is with the stimulation from a helper T cell, differentiates into a plasma cell. And the third step is that the plasma cells proliferate and produce antibodies against the antigen. Now, for cell-mediated immune system, the intracellular antigens expressed on the surface of a cell infected by a virus, bacterium, or parasite also may be expressed on surface of an APC. If you remember, I don't, so we need to go back to APC and figure that out. And then the first step is that a T cell binds to MHC antigen complexes on the surface of the infected cell, activating the T cell with its cytokine receptors. And then the second step is that a helper T cell produces cytokines that cause the activated T cell to differentiate into a cytotoxic T cell. These cytokines also influence the formation of plasma cells and activated macrophages, and then the infected target cell is lysed by the cytotoxic T cell. So there we have the differences between cell-mediated and humoral antibody-mediated immune system. We will come back to this later. But as you can see, both T cells and B cells share the helper T cell, the cytokines. The cytotoxic T cell attacks the target cell, and the infected target cell is lysed by cytotoxic T cell. But the memory B cell, some T and B cells differentiate into memory cells that respond rapidly to any secondary encounter with an antigen. So it is also T cells can differentiate into memory cells. So we have both memory T and B cells. And for this, however, we can just press hard. A type of protein interferes with viral replication and dispersion. It is the interferons that they interfere with the viral replication and dispersion. And we have so these interferons are given off by the infected cells. So the part of the adaptive immune system or humoral immunity is what is responsible for generating antibodies or to specific antigens. It should be the B cells or plasma cells. B cells are responsible for generating antibodies to specific antigens. They are part of the adaptive immune system or humoral immunity. They require stimulation from helper T cells. So the helper T cells stimulate the B cells to generate antibodies. So this is also important to keep in mind. We can see here that the first step is binding to antigens. So the B cell has a receptor and it binds to the antigen, and for this example it couldn't bind to the antigen, And then the second step is that there is a chemical signal. The helper T cell gives the signal, and that enables the B cell to become a plasma cell, and then the plasma cell is able to release antibodies. So that should do the job for now. We press hard for this. We're good. What is a blood protein produced in response to a specific antigen? So a blood protein produced in response to a specific antigen is antibody. So antibodies, we know that they are in the blood, so they are a blood protein, and they are produced in response to a specific antigen. Whenever you are saying a specific, and then you're also dealing with antigens, it means that it is antibodies, they are specific, and they are specific to the antigens that they represent and they attack. And here is a structure of antibodies. We know that they have a light chain, and then they have a heavy chain, and they have also variable regions. So these sections, end sections, with the orange circle, they are the variable regions, and then those variable regions are attached to a light chain on the left side and a light chain on the right side. So 2 light chains in total, and 2 heavy chains also in total. And there is also a constant region, and we can see that the constant region is stabilized with the heavy chain. And that's the structure for antibodies. But since we didn't get it this time, we have to do it again. What is the term used to describe mutation of the antigen binding site, variable region on an antibody? So what is the term that is used to describe mutation of the antigen binding site? The variable region on an antibody, the mutation should be hypermutation. We need a way to have this in our head. So hyper for antibody, variable region. It results in varying affinities of antibodies for a specific microbe. 5 different isotypes. We have IgM, D, G, E, A. So IgA, Ig. Do we have B? No. C? No. D? Yes. E and G and M. So A, D, A, B, C, D, E, F, G. A, D, E, G, M. 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 Okay, and the results in varying affinities of antibodies for a specific microbe. So IgM is the first antibody made against an infecting pathogen, and somatic hypermutation selects for antibodies that bind more tightly to the pathogen, and switching antibody isotope to IgG allows delivery of the pathogen to phagocytes. Okay, I need to take a break, and we'll be back right after this.